Today's lab is going to be a hemagglutination test that is used to detect antibodies that are produced in a mononucleosis infection, also known as infectious mononucleosis or IM. So in this test there are two controls. There is a positive control and a negative control. You can see here, positive and negative. So we're going to be running those today. And then I am going to be running one patient. And you can use serum, plasma, or whole blood in this kit, which is made by Rimmel, and it's called the Color Slide 2 Mononucleosis Test. Today I'm going to be using serum. Also in the kit there are two reagents. There is reagent A and reagent B. And the first thing in the instructions says to remove these white caps from the tubes and instead to place in them a dropper. So I've already done that with the set of reagents I'm going to be using from my kit today. So I have reagent A and reagent B with droppers. I'm going to mix them just a little bit, make sure they're at least partially mixed before we begin. And in this kit, we have to add reagent A, we have to add reagent B, and we have to add a drop of either control or patient sample to 3 separate areas on the reaction card. Just like when I showed you previously with RF and CRP testing, you can't add the drops directly on top of one another in the reaction card. The same is true for this test, and that's because we're actually going to mix reagents with patient sample in a very specific order in this test. And I'm going to describe why that is to you in the next video explanation of the principle. So the first thing I'm going to do is I'm going to label my card, positive control, negative control, and patient 1. I'm going to make sure reagent A is mixed well, including the reagent that may be in the dropper. And I'm going to place 1 drop of reagent A to the left side of the circle for each of the tests I'm going to perform. And it does point out to you in these instructions that you need to make sure your dropper is held directly vertical, because it's very important to get a precise, roughly 40 microliter drop from each reagent that we're mixing together so that we are in the zone of equivalence for this reaction. If you were to hold the dropper to the side, your drop will actually be larger than it's supposed to be. The next reagent we're going to add is reagent B. Make sure it's well mixed. This is the reagent that actually does contain some horse red blood cells. Reagent A is simply a reagent that contains antibodies from a guinea pig. But reagent B, you can tell it has blood in it. It's kind of a dark brownish color at this point. It's not really blood-like, but it does contain horse red blood cells. Again, I'm mixing, and I'm writing this drop directly vertical, drop to the right side of the reaction circle for positive control, negative control, and patient. Next, I'm going to make sure my positive control is mixed well. There are special dispensers that come in this kit, and it actually almost looks like a mini pipette with a bulb at the top. It's got the stirrer at the top, and it's got a very tapered, tiny tip on the end. So I'm going to use that to add a drop of well-mixed reagent A, and I'm going to add it kind of to the left side, but below and not touching the drop of reagent A. Okay. Next, I'm going to do the negative control the same way. I'm going to well-mixed, tiny drop, a little below reagent A, kind of to the left side. And lastly, my patient sample, patient 1, serum, well-mixed, little below reagent A. So the first step in mixing in this card is going to be to mix reagent A with your patient sample first. We're mixing antibody antibody that's possibly present in patient sample with antigens from proteins from a guinea pig. And so we're going to mix those together first. And then after those are well mixed, we're going to bring in reagent B with the horse red blood cells and completely fill the reaction circle with our mixture. Do the same thing for our negative control. First mix it with reagent A, then bring in reagent B with your horse red blood cells. And lastly, your patient sample. First with reagent A, mix guinea pig antigen first, then horse red blood cells. Fill the reaction circle. And this test only takes 1 minute. So I'm starting a timer for 1 minute. And I'm simply rotating the card just as we did previously with CRP and RF tests. And we're looking for visible agglutination of the red blood cells, which is why this is called heme agglutination. This is going to be a direct heme agglutination because we have not added anything to the horse red blood cells. If the antibody produced in infectious mononucleosis is present, it will bind to antigens normally present on the horse red blood cells, causing agglutination. So if we look really closely at our positive control, these clumps are not as large as those that were produced in the latex agglutination tests from Friday, but you can see as I move the positive control around, you can see the small red agglutinates. With the negative, you don't see that, so we'll try to get both in here. You can even see the clumps kind of collecting around the edges in the positive control, but that's not happening in the negative. And lo and behold, our patient is positive. They have small clumps of horse red blood cells present. So we would report positive for this patient.